Hello, biologists, and welcome to our next lesson in the biochemistry unit. Today we're going to be talking about proteins. Now remember, our four biological macromolecules, carbohydrates, proteins, lipids, and nucleic acids, are all examples of polymers, which are large molecules. And these polymers were made of monomers, which were the individual units of polymers. And today we're going to focus on proteins. Now, you've probably heard of proteins in terms of foods that are high in protein, like meats, eggs, cheese, and nuts. However, proteins are found in many more places than just food. They're found in every living organism like you and me. Our nickname for proteins is the worker bees of the cell because they have so many different functions. And the function we are most concerned with is the ability for proteins to act as enzymes. And we're going to talk about enzymes in a bit here. Now, our monomer of proteins is an amino acid, and our polymer is a polypeptide. For our elements involved, carbon, hydrogen, oxygen, and nitrogen are going to be found in all proteins. Now, sulfur here is in parentheses because this is only going to be found in some amino acids. Speaking of amino acids, let's see what they're all about. Here we can see the basic structure of all amino acids. We can see that every amino acid has an amino group and a carboxyl group. So those were two of the functional groups we talked about previously. And every amino acid will also have a hydrogen atom here and an R group, which varies depending on which amino acid we're talking about. And this R group here is what makes every amino acid unique. So some amino acids are going to be polar, some are nonpolar, some have really large R groups, like this arginine down here. And some of them have really simple R groups, like just a hydrogen atom shown in glycine. So we actually have 20 different amino acids. Now, our bodies can make 12 of these amino acids naturally, but the other 8 we have to get from eating foods that have protein, like meat, beans, and nuts. So again, our monomer of proteins was an amino acid, and amino acids link together to form a polypeptide shown here, which is the polymer of proteins. Now, a polypeptide is going to be a chain of amino acids that are linked together by peptide bonds. So I want to point out here that these peptide bonds between our amino acids are covalent bonds. And remember, covalent bonds are the sharing of electrons. And these bonds are going to be really strong and hold our amino acids together. And our polypeptides here can be formed by dehydration synthesis, which we talked about previously. So remember, in dehydration synthesis, the hydroxyl group of one molecule and the hydrogen of another molecule combine, and this releases water as a product and forms a covalent bond between two molecules. And in this case, that covalent bond that's formed is a peptide bond. Now, proteins can be made of one polypeptide chain or many polypeptide chains, such as in hemoglobin. So here we see the structure of hemoglobin, which is a protein found in your red blood cells. Now, this protein is made of 4 polypeptide chains put together, so each one is represented in a different color here. And hemoglobin is a really important protein because it helps our red blood cells carry oxygen. So now let's look at some of the other functions of proteins. One function of proteins is transport. So we have an example here of channel proteins, and these help substances cross the cell membrane. Proteins also help with defense. So our example here, antibodies. These are proteins of our immune system. Antibodies recognize pathogens like viruses, and they allow our immune system to respond and fight off these pathogens. Proteins can also function as hormones. So our protein insulin here helps regulate blood sugar. Proteins can also offer structural support. So we have an example here of keratin. This is a protein that's found in our hair, our nails. It's found in the feathers of birds and on the scales of reptiles. And this helps make these structures tough. Proteins can also help with movement. So actin and myosin here are two proteins that work together to help your muscles contract. And last but certainly not least, proteins can function as enzymes. And this is the most important function of proteins, their ability to function as enzymes. So let's look at these enzymes in a little more detail. So what exactly is an enzyme? Well, an enzyme is a biological catalyst that speeds up reactions by lowering the activation energy. So I want to look at this diagram over here. The purple line is a reaction occurring without an enzyme. So this is no enzyme. And this green line here is a reaction with an enzyme present. So we can see that in the purple line, when we don't have an enzyme, it takes a lot of energy to get the reaction started. But when we add an enzyme, shown in the green line here, our activation energy is much lower. So it takes a lot less energy to get the process going, so overall our reaction is going to speed up. Another thing I want you guys to know is that the names of all enzymes end in the suffix ase. So let's look at some examples. DNA polymerase is an enzyme that adds nucleotides to help complete a strand of DNA. Lactase is an enzyme that breaks down the sugar lactose, and we talked before how people with lactose intolerance are actually missing this enzyme lactase. And without this important enzyme, it's difficult for the body to break down the sugar found in milk, so this causes stomach aches. ATP synthase is an enzyme that produces ATP, and the name itself kind of tells us this. So the synth here tells us synthesizing. So ATP synthase makes ATP. And lastly, we have amylase here. So this is an enzyme that's actually found in your saliva, and it helps break down complex carbohydrates. So this helps start the process of digestion. And overall, enzymes are really important because they speed up the chemical reactions in the body. Now, we can't talk about enzymes without also talking about substrates. So enzymes are really picky proteins that only act on certain substrates. And we call this the lock and key model. So when you think about locks and keys, only certain keys will open certain locks. And enzymes and substrates are kind of the same way. Certain substrates fit into enzymes like a key into a lock. Now, substrates bind to enzymes at what we call the active site here. And this forms an enzyme substrate complex. Now, once this complex forms, the enzyme catalyzes the reaction and converts these substrates into products. Lastly, these products will be released from the enzyme, and the enzyme's active site is free again. So this enzyme can now receive more substrates and catalyze or speed up some more reactions.